To try and give you some idea of how these neurons are organised, and this is just taken from part of the hippocampus, but just you see, taken from a paper by Chung, which was published recently using some special techniques, you can see the beauty of these neurons. And once again, you see that the cell bodies there, these long axonal processes and dendritic trees, and when you imagine the thousands of neurons which are connecting up with this, how each one of those might regulate the inputs and outputs of this one neuron. So we have pathways, and we think, I think as Barbara pointed out, we think of often that there are single pathways. So we can think of them, a very simple one to think about is the visual system. So we have our sensory organs like the eyes here, and then pathways of neurons which then extend from those through the white matter through to the cortex. But it's really important that we realise that it's not really just a whole series of divisions. The brain isn't operating as a single set of lots of little discrete regions. We now know from a lot of the techniques, and you'll hear a lot more about this during the 2 days, the whole brain is involved in a lot of processes. If you look at language, for example, or look at hearing, which is my specialty, it's not just the regions around the cortex, the temporal cortex here, which are associated with, say, hearing and language. If you depending on the type of stimulus you provide, the whole brain, particularly the parietal lobe and parts of the occipital lobe, which is involved in vision, all will light up when you listen for or are involved in language. So it's not everything is discrete. And this might be also why we know that people who have, say, strokes or brain injury, they might have a discrete region which is affected, but they can recover other parts because some of the information and some of the processing involves so many other parts of the brain which are still healthy. And one reason why many of, we can get some recovery after some of those sorts of severe injuries. And this just to show you, I think this is a beautiful movement from the same paper by Chung, using this very, very special technique which they can load dyes into the neurons, and then they can clear the brain so it's got no tissue around it, if you like, makes it transparent, and then photograph it and zoom in around through the brain and look at all the connections. There's the connections which are involved, and in this case we'll see parts of the brain stem. This is the brain stem here, this is only just part of the brain. But all this yellow here are the nerve tracts and the cell bodies of neurons associated with in the rat brain. So you can see here's part of the cortex, all those neurons. See the fiber tracts coming up from the thalamus. And it's these sorts of techniques which are allowing us to get so much more of an understanding of how the brain connects, how all the different pathways are connected, and particularly then looking at things like plasticity, which you'll hear a lot more about later on. So here's the brainstem pathways coming in. I think it just gives you a bit of reality to that three-dimensional view of this really beautiful structure.